Hi everyone, welcome to my microbiology disease report presentation. Today we will be discussing leprosy. Leprosy is one of the oldest documented human diseases, with descriptions appearing more than 2,000 years ago in ancient civilizations such as India, China, and the Mediterranean. Historical texts, including the Bible, describe people with leprosy being isolated or sent to live apart from society. In 1873, the Norwegian physicist Gerald R. Muir-Hansen identified Mycobacterium leprae as the causative agent of leprosy. This was the first time a specific microbe was linked to a chronic human disease, helping support Robert Koch's germ theory and shifting medical understanding away from ideas of curses or hereditary causes. Throughout history, leprosy carried heavy social stigma. People with the disease were often forced into isolated communities called leprosaria. This stigma even influenced the naming. The common name is leprosy, which refers to the scaly skin lesions on a patient. The scientific name is Hansen's disease, and historically it was referred to as the separating disease or the isolating disease because of the social exclusion associated with it. M. leprae is classified under the domain Bacteria, phylum Actinobacteria, class Actinobacteria, order Mycobacterialis, family Mycobacteriaceae, and genus Mycobacterium. This places it within a group of gram-positive bacteria known for their thick, lipid-rich cell walls. Within the Mycobacteriaceae family, M. leprae is closely related to M. tuberculosis, which causes tuberculosis, and M. avium, which often infects immunocompromised patients. These relatives tend to grow faster than M. leprae and are associated with lung and wound infections. Unlike most of its relatives, M. leprae cannot be cultured on artificial media. This is due to its reduced genome, the result of reductive evolution, which caused it to lose many metabolic genes. As a result, it has an extreme dependency on host cells and exhibits very slow growth, distinguishing it from other mycobacteria. Unlike many pathogenic bacteria, M. leprae does not rely on typical virulence tools like type 3 secretion systems, AB exotoxins, or classic biofilms. However, some studies show that it may still form biofilm-like clusters in tissues or inside macrophages, which may help with survival. Instead, M. leprae expresses a unique glycolipid called PGL1, which allows it to attach specifically to Schwann cells and peripheral nerves via complement receptor 3 and laminin 2. This targeting explains why nerve damage is central to leprosy. After entering Schwann cells or macrophages, M. leprae prevents the normal cellular destruction pathway by blocking fusion of the phagosome with the lysosome. This lets the mycobacterium survive, replicate, and persist for years. The cell wall component, LAM, reduces T cell activation and helps dampen the cellular immune response. This immunosuppression allows the infection to spread and determines disease severity. Strong immunity leads to tuberculoid leprosy. Weak immunity leads to leprotomous leprosy. Because M. leprae grows extremely slowly, dividing every 12 to 14 days, the infection is chronic and persistent. The immune system forms granulomas, clusters of macrophages and immune cells, to contain the bacteria, but this can also contribute to nerve damage over time. For Mycobacterium leprae, all currently identified strains of M. leprae are pathogenic, in other words, are able to cause the disease, and there is no knowledge of any harmless strains of the Mycobacterium. M. leprae is known as an obligate pathogen, which means it cannot survive outside of a host, which will result in it causing disease upon infection. This is why all known strains are pathogenic, is because M. leprae undergoes reductive evolution. Reductive evolution is the process when an organism's genome loses genes over time. This is because when a pathogen lives within a host cell, it abandons the genes that it needed to live independently because they are no longer needed. Many lost genes are for metabolism, protein synthesis, and DNA repair, which are things M. leprae can take from its host. There are only a few strains of M. leprae, and these strains are very genetically similar. According to Oxford Academic, despite genome degradation and the presence of many pseudogenes, M. leprae strains have exceptionally low genomic variability. Originally, researchers formed a system where the strains were grouped into four main single nucleotide polymorphism, or SNP, types. However, this system was later expanded to include subtypes. Now there are around 16 to 19 genotypes of M. leprae. Examples of M. leprae strains include the 1B Bangladesh, 1D Malagasy, 3I, and 4N/O. These strains are different based on single nucleotide polymorphisms, SNPs. However, all of these strains can and will cause leprosy. These strains do not differ in their virulency or non-virulency. These strains can be found in all different places of the world. In Asia, the most common strains of M. leprae are SNP type 1. In Africa, mostly SNP types 2 and 4. Historically, Europe has the presence of strains SNP types 2 and 3. Lastly, in the Americas, the SNP type 3 is most common, and the subtype 3I can be found in armadillos. Overall, all M. leprae strains are pathogenic and have little to no genetic variation. The strains are named following the SNP system, ranging from types 1 through 4, along with subtypes. And while the strains don't differ from virulence, their distributions throughout the world make them distinct from each other. Mycobacterium leprae is a rod-shaped bacterium or bacillus-shaped. It is an acid-fast bacterium and thus gram-positive. Acid-fast bacteria are gram-positive and will have a thick peptidoglycan layer. Since it is acid-fast, this means it does not stain well with gram stain because of its waxy mycolic acid-rich cell wall. Since it's an intracellular bacterium, it means it cannot be grown on artificial media and only grows inside host cells. M. leprae's slow-growing property makes it one of the slowest of all pathogenic bacteria to grow, about 12 to 14 days. It has no flagella, and its cell wall has peptidoglycan along with unique glycolipids, specifically phenolic glycolipid 1, that allow it to avoid the immune system's response to it. Its aerobic property means that it requires oxygen to survive, and its mesophilic properties means that it thrives at human body temperature. Leprosy is most easily identified by its unique skin lesions and loss of sensation, which are the key hallmarks of the disease. The hallmark of leprosy is a skin lesion that becomes numb. This loss of sensation occurs because Mycobacterium leprae attacks peripheral nerves, especially those near the skin surface. Unlike other infections, the lesions are typically painless, which is a major sign for clinicians. Leprosy lesions can vary in color depending on the patient's skin tone and how their immune system reacts. Some lesions can appear reddish, while others appear hypopigmented, which is lighter than normal skin. The hallmark image included here from the report shows a person's thigh with many lesions of different sizes. This variability helps clinicians distinguish leprosy from conditions that produce uniform rashes. The lesions often have white, dry, and scaly edges. This happens because nerve damage reduces sweating in that area, leading to dryness and flaking. These scaly edges are visually distinctive and help confirm diagnosis. The term leprosy comes from the Greek word lepis, meaning scale, and lepo, meaning to peel off. The dry, scaly appearance of these lesions is the historical reason the disease received this name. Leprosy is a chronic bacterial disease with a long incubation period, and it does not usually cause a high fever, a sore throat, pustules, or a rash in ways that acute infections do. Symptoms of leprosy include painless skin lesions, peripheral nerve damage, which can cause numbness, tingling, weakness, and paralysis in the hands, feet, or face, and deformities. Leprosy's incubation period can range from 2 to 10 years, and the symptoms will worsen slowly over months to years without treatment, as leprosy is a chronic and progressive disease. The contagious period, when a person begins multi-drug therapy, or MDT, ends within days to weeks. Leprosy is not easily transmitted. It typically spreads through extended, close contact with someone who is untreated. This long-term exposure is why household contacts, rather than casual acquaintances, are more often affected. The primary mode of transmission is through respiratory droplets released when an infected person coughs or sneezes. These droplets can enter through the nasal passages or respiratory tract. making it similar to the spread of other airborne or droplet-based infections. Quick, everyday interactions, such as shaking hands, hugging, or sitting near someone, rarely transmit leprosy. Infection usually requires prolonged and repeated contact with someone who has active, untreated disease. Direct contact with infectious skin lesions or contaminated environmental materials like soil or water where M. leprae is present can transmit the disease, but this route is much less common than respiratory droplets. Entry through broken skin is required for this route. In parts of the southern United States and central South America, 9-banded armadillos are natural reservoirs of M. leprae. Handling or having close contact with infected armadillos can result in zoonotic transmission to humans. M. leprae primarily infects skin macrophages and Schwann cells in peripheral nerves. These infected cells present bacterial antigens to T cells, beginning the immune response in both skin lesions and nerve tissue. There are two ways the immune system responds: by activating cellular and humoral immunity. Cellular immunity, which is T cell driven, and humoral immunity, which is antibody driven. The balance between these two immunities determines disease severity. There are two forms of leprosy: TT, which is tuberculoid leprosy, and LL, which is leprotomous leprosy. Tuberculoid leprosy is dominated by a strong cellular response. Leprotomous leprosy is dominated by antibodies and a weak cellular response. Tuberculoid leprosy has a strong T cell response, which controls infection. In tuberculoid leprosy, a strong T cell response activates macrophages and keeps the bacteria contained. This leads to few lesions, minimal bacterial load, and non-contagious disease. Leprotomous leprosy has a weak cellular response, resulting in many lesions and high numbers of antibodies. In leprotomous leprosy, the cellular response is weak. The body produces many antibodies, especially against PGL1, LAM, and heat shock proteins, but they don't stop the bacteria. This form causes widespread lesions, high bacterial load, and is contagious. There is no specific leprosy vaccine that provides complete protection nor is widely used. However, the Bacille Calmette Guerin, or BCG vaccine, which was originally made for tuberculosis, provides partial protection against Mycobacterium leprae, as studies show that it can reduce the risk of leprosy by about 20 to 60%. Since M. leprae is not transmitted through contaminated water, leprosy is not a disease brought out by contaminated water. Therefore, providing a clean water supply does not directly reduce infection. Hand washing also doesn't directly reduce infection, as leprosy is spread through prolonged close contact with an untreated person rather than through casual contact. In other words, leprosy is spread through human interaction along with armadillos in certain regions of the Americas. Public health campaigns are effective ways to directly reduce the spread of leprosy, as their approaches include early detection, contact tracing, health education, and free multi-drug therapy treatment. Overall, leprosy prevention is brought about by the approaches offered by public health campaigns, and the BCG vaccine offers some protection, but there is no specific or widely used leprosy vaccine as of now. Diagnosis of leprosy starts with recognizing the characteristic symptoms of leprosy and then confirming them using laboratory tests. Clinicians will combine visual inspection, sensory testing, and microbiological methods. There are key signs which are observed. According to the World Health Organization, the two most important clues are painless, numb skin lesions that don't respond to touch, pain, or temperature, and thickened peripheral nerves, sometimes tender to the touch. These reflect nerve damage caused by M. leprae. Skin smear microscopy can detect acid-fast bacilli, or AFB, in slit skin samples. A skin biopsy provides histopathological evidence of M. leprae and helps determine the type of leprosy. This is one of the most reliable clinical tools. The leprotomin test is not used to diagnose leprosy. Instead, it helps classify the disease into tuberculoid or leprotomous leprosy based on the immune response. Included in this slide are the references that I used to make this microbiology disease report presentation. Thank you for listening to my presentation and have a good day.